In 2014, the University of Wyoming hired Dr. Holly Ernest as the first holder of the Wyoming Excellence Chair in Disease Ecology, a chair founded with the help of the Wyoming legislature. Upon arrival, she began to head up its wildlife genomics and disease ecology lab. Her training and research has led her to describe herself as a, quote, wildlife biologist, population geneticist, and a research veterinarian. What in the world does that mean? Wildlife biologist. That's probably the easiest. Holly studies Wyoming's wild animals. In particular, those we like to see, the photogenic megafauna like black bears, mountain lions, and bighorn sheep. She also studies less attractive critters, ticks, bugs, germs. Oh, and she's also an expert on Wyoming's ubiquitous magpies, as well as hummingbirds, and as we'll hear today, owls. Research veterinarian. Well, I think we can figure that one out. Vets deal with health and diseases of animals, and research means that she studies those diseases. Okay, all right. But it's the term population geneticist that brings it all home. Dr. Ernst looks at the genetic makeup of and variation in wildlife populations. In particular, she is interested in how genetic variation changes over time, especially under the impact of pressures from diseases in human population. Dr. Ernst earned her Bachelor's of Science from Cornell University, and then in 1986, her Doctorate of Veterinary Medicine from Ohio State, and followed that several years later with a PhD in ecology with an emphasis on wildlife genetics and population health. She taught at UC Davis for 13 years before coming to UW, where she teaches, quote, students to grow their appreciation and talents in the art and science of designing and conducting studies involving DNA, ecology, and population health analysis of free-ranging wildlife. We've got a lot of free-ranging wildlife in Wyoming. Now, I'm not quite sure which animal she's talking about today, but I am looking forward to hearing her speak on genomic testing, good for humans, good for wildlife. Dr. Ernst.
Thank you, Paul. So wildlife that you live with around your neighborhood, they breed, they eat, they're preyed upon, often without you knowing a thing about them. You might see them for a millisecond out of their life. And so the science that I do, I was drawn to it by trying to learn more about them when we see such little parts of their lives. So I also watch TV at night, often Law and Order. I watch that a lot. And during that time, probably many of you see advertisements for 23andMe and those other genetics companies. And so I thought I'd frame my discussion with you today around that and compare and contrast those 23andMe genomic companies that take a little bit of your DNA when you want to send $99 and then test where you came from, what your relationship is, maybe your ancient history. Now on the left-hand side is a drawing from 1500s. It's a woodcut of a pedigree of a man. And so he's down at the bottom. It's hard to see from there, but there he is at the bottom with all his ancestors. Back at that time, we could only go on written record, and sometimes not even that. But now we have our 23 sets of chromosomes, and that's where the company 23andMe comes from, to look at, to test our ancestry. Now, there are precautionary principles with this. There's some criticism with these companies, Ancestry.com, 23andMe, and others, is that they're not always right. And so you do have to be buyer beware. They base testing your DNA on a reference data set. It's getting better and better, but early on it was kind of crappy. And so that's one thing I'll say is that some of the work I do is analogous, but I really hope that we do good work and that we live up to something a little better than what some of these companies do. So some of the questions that my students and I ask are, who are these wildlife really? And where do they come from? Who are they related to? And if they have a disease problem, is it related to who they're related to, or is it some genetic problem? DNA can tell us a lot. So that's what I hope I can introduce you to, is some of the concepts with what we use for DNA, some of the terminology, and then you'll go home with some new words maybe. And so I'll introduce these concepts through 3 species that we study. One is great gray owls and the Tetons. My student Beth studies them. Another is our very own Bighorn sheep in Wyoming, and a postdoc working with me, postdoctoral researcher Sierra, is studying them. And then the mountain lions that I've studied along with a number of students for the last 25 years. So first for great gray owls. These are really awesome animals. They're the biggest in dimension of all owls on Earth. Great horned owls may weigh more, but the great gray, our very own in the Tetons, is bigger. And it has what's called a circumboreal distribution. And what that means is around the globe, there's these special forests, the boreal forests, also called the taiga in Russia, that are mostly conifers, pine trees, spruces. And these compose almost 30% of the forests in the world. And they're second only to the oceans in ecosystem. They're amazing. We don't know a lot about them because they're kind of to the north. They're mostly in Russia and Canada. But our great gray owls, that's where they live. And they're at the very extent, the southern extent of their range with us in Wyoming, and I'll tell you a little bit about the California population. They're really cool because they have a number of adaptations. They've evolved to live very well in these circumboreal forests. So they have these asymmetrical ears that help funnel in the sound. They have more light receptors in their eyes, so they can really see things at night. As you know, they're night species. They have those talons to pick up those little rodents, that poor little guy that got caught and munched by that great gray owl. And they have those special dense downy feathers that help insulate them in these cold habitats. So here's your first word, panmixia. So one question that we have is, how can we tell how populations are? Are they one big interbreeding population? That's what panmixia means, all interbreeding. Or are they sub-pockets, are they pockets of animals that are distinct, that we have to pay attention to as conservation biologists? So here's some of their distribution in red on this green map, with the southernmost population that I studied in California before I came here. And so one question we might have is, do these circles represent their different populations? It's roughly over the geography where they are. Are those unique interbreeding groups, or is it one big interbreeding group? We have concerns for these owls in California as we do in the Tetons. So they're in this tiny little population in the Sierra Nevada. So this is the state of California right here. And our population in the Tetons is over here in Jackson. So these little tiny green spots that I'll point out are where great gray owls are. They're very disjunct. So this tiny little green spot in California is where the great gray owls are there now. So there's something between 100 and 200 of them. That's a tiny number. So we're really worried about them starting about 10 years ago when I started to study them. So they're in Yosemite, another Y national park like our own Yellowstone. And there's a number of threats that attack them. So one disease that attacks them is trichomonas. So that's a nasty disease that infects their throat and their gullet so they can't eat so well anymore. It affects a number of raptors. We can get a type of trichomonas, cattle get trichomonas. It's a nasty bug. Also, they're roadkill. So when great gray owls hunt at night, they often fly low over roads, and that's when they get hit. You know, the person driving the car can't see them, so they get killed by cars. And then poaching. So people may want something from a great gray owl, maybe a feather or something illegally. West Nile virus has been a concern. That was one thing that I was really interested in as I started my faculty job at UC Davis in 2001, 2002. You here in Wyoming were starting to get West Nile virus in your horses and your mosquitoes and your people. We were watching it from the west, seeing it come across the continent, And I actually started one of my major research programs just on that observation, that when I rode my bike into work, I'd notice the Swainson's hawks. We have them here. They're awesome. And the magpies, yellow-billed magpies there, black-billed magpies here. And owls that were getting hammered in the Midwest by West Nile virus. And so I posed the question, what's going to happen to our wildlife when it arrives? And it turns out that some of the studies in Minnesota indicated that great gray owls were exquisitely sensitive to West Nile virus, that once exposed to the mosquitoes that carry them, they would die right away. And so, man, there's only 100 or 200 left in Yosemite and the Sierra Nevada. What's going to happen to them? So with others, we started a research program to study their exposure to it. And then the question came, well, are they unique? Are they genetically unique? And actually, that study ended up to be more wide-ranging than the West Nile study. So it turns out those birds are really lucky. They're at a higher elevation, like we are at Laramie. We're at 7,200 feet at Laramie. We still get West Nile virus in our mosquitoes. Those guys are a little higher. And there's this elevational gradient for West Nile virus, above which, assuming no more climate change, that mosquitoes can't amplify the virus in their bodies. And so the great grouse in Yosemite don't tend to get it yet. Now, as climate does change and the temperature increases going up in elevation, we'll worry about them. But right now, they haven't been hit yet. So there's big fires. On the news, you may see about some of the big fires in California. This is from 2013, so some 5 years ago, and that was called the Rim Fire, and that was right in the great gray owl's habitat. So you can see there's a number of threats hammering them like other wildlife species. So one thing we wonder about, well, if they get hammered by each of these threats, it's called part of an extinction vortex, where a species might start up here in population numbers as being okay, lots of them, and then there's maybe disease, and then they get hit by cars, and then other things keep hammering them. Is their genetic diversity going to go so low that they can't exist anymore? So there's a number of things we can do using genetics, and without harming the animal very much to get that sample, to test some of these things. So one of the things we can do with genetics is actually to count individuals without having to sample them. So we can use their poop and collect a DNA sample from their poop and say, that's a unique mountain lion or a pronghorn. And then we can count up the unique individuals we have. That's also part of wildlife forensics. I'm actually a forensic consultant for the federal government to help develop new protocols for wildlife forensic techniques. We can count family units. We can find, okay, who's related to whom? We can reconstruct pedigrees like that 1500s woodcut that I showed you in the first slide. Very importantly for conservation, we can define what a population is. So what is an interbreeding, a panmictic group of animals? And so that's really important. And that helps us delineate units of conservation. And we can determine, if we know what units we have, how much interchange is there between them. So one question that we have for pronghorn, the antelope of our state, is I-80 cutting them off? So I have a PhD student, Melanie, who's working with me to examine the genetic diversity of pronghorn above I-80 and below I-80. There's a lot more questions than that, but that's looking at gene flow, genetic interchange among populations. Evaluate the genetic diversity. Is the genetic diversity getting so low that they're going to now have genetic defects? Florida panther is our classic example of that. That's a mountain lion in Florida. And their genetic diversity got so low because their populations dipped to 20 to 40 individuals where they were interbreeding with each other, that then they developed genetic defects like holes in their hearts, abnormal sperm. On the outside of them, they had these whorls of fur at the base of their tail. They had kinked tails. And interestingly, there's a population in southern Florida that has some kinked tails. Those individuals that I tested had the lowest genetic diversity. Maybe they broke their tails, we don't know, but by coincidence, these two individuals that we documented with kinked tails also had the lowest genetic diversity of all the animals we studied in Southern California. It's just a mention of precaution that we have to watch these things. We can also calculate an index of their population size. So how many individuals are there? We can use genetics as a way to count the numbers of individuals that are contributing to a gene pool. And we can examine their past history. So that's called demography, or examining long ago, did they have a genetic bottleneck? So at one time, back hundreds of years ago, was there a constriction of the numbers of individuals? Sea otters, so we're studying southern sea otters in California. During the fur trade in the 1800s, their populations were constricted, and we can actually see a mention of that in their DNA. So we can compare our known history of wildlife species, compare it with the genetic history, and draw inferences from that. So here's more of your lessons today. So it really is an alphabet soup when we talk about DNA terms. And so one of the first things that I want you to learn about are Snips. And so that's SNP, so single nucleotide polymorphisms. When we talk about genomics, like when you send your DNA off to 23andMe, they examine your Snips. It's a single nucleotide, a single base pair of the DNA that is different from others. And so that's really powerful for testing in wildlife too. There's mitochondrial DNA. It's a tiny loop of DNA that's in every cell at about 1,000 copy number, and it's our mother lineage. When we have a child, our mothers carry that mitochondrial down through generations. So it's a really powerful DNA tool that tells us about the matriline. There's also Y chromosomes. We can test patriline too. STRs, or microsatellites. This is what pioneered forensic genetics for humans. So some of the early cases in human forensics used STRs, and they're still being used. Just 2 weeks ago, I was at the federal conference of over 600 of us forensic scientists that met in Chicago. And the human geneticists still use STRs. They're starting to use Snips, but it's still a workhorse of genetics for wildlife and for humans. And next-gen sequencing, or NGS. So that's using the whole genome, including some of these Snips that I mentioned, to study genetics. So I just wanted to show you one type of DNA and how it varies in our genome and in wildlife genomes. So this is a microsatellite. And there's 3 different versions of it in 3 different animals. It turns out these are mountain lions as examples. And these are the base pairs, each letter. There's 4 letters of base pairs in our DNA, A, C, G, and T. They're different chemicals. And so in the middle here, you can see CA, 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 and there's 8 repeats of that CA. The next mountain lion has 9 repeats, and then this lion has 7. And so we can measure those lengths of DNA as a very easy way to analyze what their genetic makeup is. And it turns out these mutate or change very rapidly over time. Now, rapidly on a geneticist term is like 1 in 1,000 generations, whereas that's relatively fast. It doesn't sound fast to you. But over time, we can measure the differences across individuals using a panel of these microsatellites. So one way to visualize, okay, how big is a microsatellite, is to examine our genome. We have about 3,000 books of genome, and I'll show you how we can walk through that, thinking about your own genome. So each of our, if we think of those A, C, Gs, and Ts as letters, so each of our genome books has about 300 pages and about 3,500 characters per page. Each of those characters is one of those A, C, G, and T chemicals that I mentioned to you. And one book is about 1 million base pairs of those A, C, Gs, and Ts. The DNA content of one person is about 3 billion of these letters. And then one protein gene, so our genes, our genetic makeup makes these proteins. So one protein gene is about 28 pages out of that library. The mitochondrial genome, that mother lineage, is about 5 pages. It's a tiny little loop of DNA. And then one microsatellite is only about a sentence, a short sentence, so about 100 to 300 base pairs. That makes it really robust for forensics because it's a tiny little space that you can get out of a blood spot or a fecal sample or one case that I worked on at UC Davis was urine on a tire from a dog that helped link a purported rapist with the dog owned by the person who was raped. So getting urine out of, or getting DNA out of urine from a tire. We can use these markers. So one way that our wildlife genetics is not like 23andMe is because we have to develop these tools for each of our species. And so, 23andMe just deals with one species. In my lab, we do deal with at least 10. And, there's tons of wildlife species. So that's one difference. It takes a lot of genetic tool development for each of our species. So the hummingbirds we study, we're going to start studying bats, which I'm really excited about. It takes a lot of work to get these DNA markers or tools up and going in the lab. And we have to develop these databases for each of these species and even each of these populations. Whereas 23andMe has one set of species to develop and build their database, we have to build it each time. And then we have to invest in the funds for the bioinformatics. These days with genomics, we're working with terabytes of data. It's huge. It's mind-boggling. And so that takes a lot of expertise. Even though it might be only $99 for 23andMe for you to send a sample, for the equivalent for us, it's a year or 3 years of a highly trained postdoctoral student to analyze the wildlife data. So getting back to the great grails, I sidetracked a little bit. So Josh Hull is now one of the heads of the U.S. Fish and Wildlife Service in California, but he was a PhD student and postdoc with me, and he studied the great gray owls in Yosemite in California. And so he had to first develop the DNA panel that I led you through that process. And we had to, before we started taking blood samples or handling great gray owls, we had to practice on chickens. And so here's one of my lab managers, and Josh's dad, Buzz Hull, who was a leader in the Golden Gate Raptor Observatory, with a chicken. We harmed no chickens, but we did practice on them, taking blood. So here's now some data. It's from years of work, but if we imagine that each column, here you can see one column that's kind of pink there, represents one great gray owl. So, and then we line them up by their location, where the sample was taken. And then we use their genetics compiled into kind of one set of data represented by color. And you can see that great gray owls are not panmictic across their North American range. You can see that there's pink genetics and red genetics, all kinds. And so there's the Sierra Nevada samples in the yellow. We see a few other, genetic reds coming in. That may be a migrant. Maybe it dispersed from Idaho. And so that started to tell us there are a lot of differences in great gray owls, and those great gray owls in Yosemite are distinct. In fact, later Josh wrote a paper with other people who studied other layers of data, their behavior, their biology, other aspects of their nesting, and determined that they're actually their own subspecies in Yosemite. So next we had to think about these great gray owls' genetic diversity. So in Yosemite, they definitely have low genetic diversity compared to those to the north. And so we worry about these bottlenecks. If we start long ago with lots of great gray owls and their genetic diversity represented by all these skittles here, and then it goes through a contraction of their size and their population, then we have to think about what's going to go on there. If we do something like that, we lose some of the genetic diversity. You can see in this contraction, we only have the blue and the red genetics coming through in this analogy. And so then we're left, even if they breed back out again, we're only left with this small gene pool. Some examples of that are elephant seals along the California coast, or tule elk in California. Both of those populations constricted to this many individuals. And so you can imagine they only had maybe blue genetics or blue and red genetics. And even if they blossom back up, tule elk in California have thousands, elephant seals along the coast have thousands, they still have that low genetic diversity. Maybe they're okay on the short term, but if you go over tens of years, what's going to happen with their ability to fight diseases? They don't have that genetic diversity in their immune system to fight diseases on the long term. So that's a concern. So back to our own great gray owls right here, just north of here in the Tetons. So what's the story with them? So here they are back at the southern extent of their range, a finger coming down from where there's lots of them up to the north. And so Beth is studying those genetics. And so I wanted to mention that she's just analyzed, again, terabytes of data. She's actually doing whole genome work on the work that Josh did with fewer DNA markers and finding some interesting findings on that. And I'm not going to say anything yet about Beth's data because that'll be a future talk. But what we're finding is some interesting genetics. So next I want to move to our own bighorn sheep. So in bighorn sheep, we have a statewide assessment of, again, are they all panmictic? Are they all interbreeding? What's their genetic status? How are they doing? They are hammered by diseases that are spread primarily by domestic sheep. Domestic sheep carry Pasteurella family of bacteria, mycoplasmas, without harm to the domestic sheep. But often when a bighorn sheep goes nose to nose with those domestic sheep, they get that bug transmitted to them. Their immune systems have not been exposed to it before, and so they often bring it back to their own flocks of bighorn sheep, and with catastrophic consequences where many bighorn sheep can die. So a purpose, and this is Sierra's work, is to assess the genetic diversity of our own Wyoming bighorn sheep. And so some of the questions might be similar and some are different from the great gray owls. So one difference is, do the hunt areas that Game and Fish, our collaborators, represent on a map, represent the genetics? So that could make a difference in how Wyoming Game and Fish wants to manage them if there's different genetic health. How many individuals are in a population, that effective population size? Are the populations healthy and stable? So what's a sustainable level of harvest? Wyoming Game and Fish works really hard at this, but giving them genetic data will allow them to do an even better job. And what's the effect of translocation, moving bighorn sheep from Oregon, from Idaho, which happened over the last 40 to 50 years? Are those Oregon and Idaho and Montana bighorn sheep that were brought in, is their genetic signature still there? Are the ancestors, the descendants of those animals still living? How does that affect the immune systems of these animals that have to fight this Pasteurella pneumonia? And how are these sheep moving on the landscape? Genetics can help show us that, just like radio collars can, in a different way. And a big question that Wyoming Game and Fish and others have is rams wander a lot, and often they'll wander into a domestic sheep flock. A bighorn sheep ram doesn't care if it's a bighorn sheep ewe or a domestic ewe. They're happy regardless, and so what they do is they get exposed, and then they wander back to those innocent bighorn sheep ewes and expose them to the pneumonia pathogens. So where are those wandering rams come from? Are there some populations that are more problematic in their wandering rams than others? And poached individuals. So there's a great forensic lab in Wyoming Game and Fish. It's right in Laramie, co-located with our own university. And so we work with them a lot to develop the DNA markers to help them do their job even better. That's a pronghorn, but that's at a hunt station where we collect a lot of our samples with the help of Wyoming Game and Fish and hunters themselves who send in their samples. Usually a little piece of meat or a horn core. So when a hunter kills a bighorn sheep on a permit, hunting permit, they have to go to a hunter check station. Game and Fish drills out a core in that horn on that bighorn sheep's head and implants a passive transmitter as a marker to help prevent poaching and to be able to track these bighorn sheep heads to make sure they're legal. We get that horn dust and actually work hard to extract DNA from it, from those bighorn sheep. And there's planned captures. So the Migration Initiative at the university and Matt Kaufman's group, Kevin Monteith's group at the university do a lot of captures with Wyoming Game and Fish. We get blood samples from them. And that's all the different blood samples we get in from our our lab. That's Eric and Sierra. Eric works on Bighorn sheep and sea otters, and all kinds of different ways that we get those samples in. I mentioned before, fecal samples, horn cores, blood samples, we extract from it all. And so again, we use these different tools of genetics to help mark these different ways that we can study the population genetics and give information for conservation, using the mitochondrial DNA, the Y chromosome, and nuclear DNA, the main part of the DNA. So a little bit more about mitochondrial DNA. That can help us with bighorn sheep reach farther back in history. So the demographic history, hundreds of years ago, and sometimes even thousands of years ago, they'll have a genetic signature in their matriline, their mother lineage of mitochondrial DNA. The Y chromosome is that ram lineage, and so that can tell us how rams move about the landscape and affect the population genetics of our bands of ewes or our populations. And then these markers of nuclear DNA, the biggest part of the DNA of all of us, either the microsatellites or the genomics, the whole genomic markers. And in our lab, we're looking at all of them because in the past, we hadn't had a way to compare them and then to see which one works best. And so for preliminary findings from what Sierra found is that these bighorn sheep do have very distinct heritages throughout the state. Each color represents a different genetic cluster. So this is our state. We're over here in Rock Springs, and you can see there's some colors that match up. So Ferris Semino down here matches up with Devil's Canyon. Good thing, because that's where they were translocated from. There wasn't a population recently in Ferris Semino. Game and Fish moved them down there, and their genetics matches that. Also, there's been translocations from Darby and Whiskey Mountain over to Laramie Peak, and you can see their genetics follows that. Now here in the Absarokas and ranges just to the east and to the south of Yellowstone, you can see there's a purple lineage and a green lineage. Now it's not like there's a line in the sand there. There's actually a gradation. So this is a very coarse look. But we can see that to the north of that population, they are different than to the south. And then we can see some very specific genetic lineages over here in the Tetons around Jackson, and then also Elk Mountain. So that helps Wyoming Game and Fish and the rest of us know what's going on with our bighorn sheep. So next steps that we as scientists, as a community of population geneticists will want to look at is now that we know some of the population structure, what does that mean for their immunogenetics in relation to these terrible pneumonia diseases that they get? And are diseased herds versus undiseased herds genetically different? We don't know that yet. So that's the next phase that we want to work on. And are there some individuals that get disease more often because they're inbred? Or doesn't it matter? And are more diverse, genetically diverse herds more resistant to these diseases? Now, those of us that are geneticists that have been working in this for a while know that we're just one layer. So the genetics layer has to be put in with the habitat biology, the general movement ecology of these animals, their disease patterns, their demography, their evolution. So it's one layer in their ecology and their conservation. And I think, as I've learned from working with so many people in Wyoming, is that we all have a common goal. We work a lot with the hunters, with the Wyoming Wild Sheep Foundation and others, and it's to make sure we have healthy populations of bighorn sheep throughout Wyoming. So before I finish, I wanted to switch to one more species, and that is mountain lions. And this is P22. He's also known as the Hollywood lion. And he's an interesting guy. He has a website, and he has a Twitter account. No, he doesn't tweet, but somebody tweets for him. And he's, as far as I just checked online to see if he's still alive. I've tested his DNA, I've put him in a pedigree, and I think he's still alive. He has a movie about him. So on his Facebook account, people follow what he's done. So he's interesting because this is a real picture. There's a Hollywood sign behind him. It says Hollywood right there. He's a radio-colored lion. He has a radio collar on this picture. And he lives right in the center of Los Angeles, right in Griffith Park. He's lived there for years. It's amazing. He's gotten into some trouble. He jumped into the LA Zoo and killed a couple koala bears. And we know that because the telemetry signs, saw him going in over the fence. And then there were the koala bears missing and a little bit of koala bear fur. And then the fecal samples left behind kind of DNA matched with what we know about his DNA. He also has killed llamas. He hasn't hurt any people yet. It's amazing. He lives in LA for years, and he has not harmed a person. People are afraid because he kills these other things. But he is kind of one person, or one mountain lion that Angelinos follow. They love P22, the Hollywood lion. So mountain lions in California, before I came here, it's pretty awesome to compare wildlife in the most populous state and a very blue state with Wyoming, the least populous state, where at times we've had more pronghorn than we have had people. And I love the comparisons. I still do a lot of work in California. But mountain lions do have a challenge living there, as you can see from P22's example. It's a habitat mosaic. There's some lovely areas that are just like Wyoming in the Eastern Sierra, but then there's those ones that live near LA. So there's the coast regions. Mountain lions, that's their habitat, but people are building homes like crazy along the coast, all up and down. And so there's incredible pressures that our mountain lions face there. And then we have these different areas that have differences in how mountain lions are affected. There's the pot farms in the Mendocino area, the North Coast Range, where there's a lot of poisons put out by illegal pot farms. And so the mountain lions have been poisoned by a number of problems there. There's the wildfires, like I mentioned, in the Sierra Nevada for the great gray owls. That's harmed mountain lions and other mortality sources. Here is I-15 that goes from Los Angeles to San Diego. It's a 10-lane highway. There's two populations that are split by that highway. So some of the objectives I had when I started my PhD there some 25 years ago, and I'm still examining, is to determine their genetic measures of their health. How are they doing? And to determine what is a population of mountain lions. When I started out, California Department of Fish and Game at the time, now Fish and Wildlife, kind of laughed at me and said, "Fine, little girl, they all interbreed. They're not different." I found otherwise. And so also, we want to evaluate disease impacts. We're working on an NSF, National Science Foundation grant, with Colorado State University to examine the differences between genetics and their diseases. And to find measures that we could track over time to help the wildlife agencies monitor how they're doing. And so some of the populations that I examined with my students was the Santa Monica Mountains and the Santa Ana Mountains. In between there is Los Angeles, and that right there is where P22 lives. So here's some of the habitat that they live in, and zooming in, again, here's LA. There's I-15, that 10-lane highway, and the Santa Monica Mountains is right in there. Ventura County is right there. The Santa Anas is right here. There's a big Army base there for good because that's preserved a lot of their habitat there. I-15 runs right between these two populations. So this other population is to the east in the Eastern Peninsula Range. And so one thing that I was able to do over my years in collaborating with National Park Service and others is over a 15-year study of mountain lions that were captured for radio collaring is to use their DNA to reconstruct their pedigrees, starting with P1 here. And our Hollywood lion places right there. The other thing we could do is, again, using that same genetic analysis where we assign colors to different genetic groups. Each column is a different mountain lion. As you can see, there's a green population of mountain lions, a blue one, and a red one in these areas around Los Angeles. And so here is where P12 came in, and he actually introduced new genetic diversity quite naturally into the Santa Monica lions. And you can see how there's more blues and greens. That represents new, fresh genetic material. So that was interesting for that population. So the landscape in California and here is changing rapidly. And so one question we have is, how is that affecting their populations on a genetic standpoint? And will it help to put in wildlife overpasses? People are starting to talk about that for I-80. Is that important? It costs millions of dollars to do that. Is that wise use of money? I'm just asking you questions so that you can think about it when you vote for these kind of things. Here's a picture of one of the mountain lions that I studied in the Santa Anas, M89 with her cub. Similarly, I was able to reconstruct the pedigrees of those animals down farther to the south and assess that I-15 is having a huge effect, that 10-lane highway, on the differences in these mountain lion populations. And so my message to you is with these 3 examples, hopefully you're seeing how genetics can give us more information than we had before on these animals. There's a lot of uses for DNA. We have problems just like 23andMe does. We have to develop good reference databases, garbage in, garbage out if we don't have good genetic databases. We're using them to answer questions for conservation. We're developing new genomic tools, and hopefully we have applications for conservation. I thank you very much for listening.
With the North American bison population going from millions down to, I guess it was a few hundred maybe, and then I guess it's back on the rise now, but did that low genetic diversity, is that why they're so susceptible to like brucellosis and stuff?
So the question is about bison, you know, again, one of our Wyoming heritage animals, going from millions across the plains and elsewhere to a few thousand. Is it a problem for genetic diversity, and is it a problem for brucellosis? A number of questions. Those are great questions. Thank you. So yeah, they were really bottlenecked, genetically bottlenecked. We don't know the extent to which they're going to suffer from this low genetic diversity. People are just starting to study it. There is a problem. With brucellosis, so my husband is also a veterinarian, has a PhD in epidemiology. He runs the brucellosis program for the agriculture school at University of Wyoming. So I hear a lot about brucellosis from him. And it turns out that even though bison are blamed a lot for brucellosis, the bigger threat for cattle ranching is most likely elk. So they're wider ranging, they're expanding their populations, and they're more likely to transmit brucellosis across the landscape into our beef cattle herds, which could shut down our beef cattle industry, than bison. Now, bison can still carry brucellosis. We don't know yet. Your original question was whether their contraction of genetic diversity will cause them to be more susceptible. We don't know that yet because we don't have the genetic information to tell us. You know what might be more important there is the exposure. So I think keeping bison separate when we can, quarantining them when we translocate them to another area, like many places are trying to do, and to be careful with how we interact bison, elk, and cattle.
Yeah, I was just wondering, you always hear about, you know, genetic engineering and cloning and so forth. It sounds like you guys are developing these big DNA databases. I'm just wondering what the current thinking or even ability is to genetically manipulate some of these and make them more resistant to disease or something like that, like with the owl and the sheep population. I mean, is that getting any thought, or what's the current thinking about that?
Yeah, that's another great question. So the question is what about cloning and genetic engineering for wildlife conservation? What's the status there? People are attempting to do it. Researchers are attempting to do cloning of extinct animals. People are working on trying to resurrect mammoths or Tasmanian, not Tasmanian devils, but there's a Tasmanian dog that went extinct. A marsupial, not like our own mammal dog. But the question some of us conservation biologists have is, what good does that do if they don't have their habitat? So there's an ethical, moral question going on that I could see with the philosophy talk earlier on. We have to think these things through. So there's getting to be the ability to do these things, but will it help wildlife? So there was just in the past couple of days, there was a piece in one of the Wyoming newspapers about controlling the genetics of our mule deer to help reduce the risk for chronic wasting disease, that prion disease that's spread by proteins, not germs. And so I think we really have to think that through too, because when we change the genetics for one little disease, we don't know what we're carrying along that may be good or bad when we select for that resistance for chronic wasting, we may be inadvertently selecting for something bad. So this is where a lot of thought, a lot of community discussion is really important. So thank you for that question.
The ability?
I think the ability is...
Genetically engineer that kind, I mean, if you wanted to, science is to the point where they could do it. It's just the question still remains, should we?
Should we? It's getting there. You know, there's Dolly, the sheep that was cloned some 10 or 15 years ago. Now each species, just like I mentioned how 23andMe only deals with humans, we deal with all these different wildlife species. It's the same thing where if you're going to try to resurrect cloning or genetic engineering, you have to specialize it for each species. So we may not have it for each wildlife species, but the science is getting there. And so I think we as a community, as a society, have to decide, is this something good or not?
You talk about those bottlenecks. Have you done any work with grizzly bears? Like how their population shrank and now it's grown? Is there any studies on that?
There's been tons of work. The question is, have there been studies on grizzly bears? And so I don't work with grizzly bears. I work with black bears. But there have been a lot of research. There's a University of Idaho researcher, Lissette Waits, who's done lots of work on grizzly bears. And so that'll be really important as our Endangered Species Act designation for grizzly bears in this state is changing. It's becoming a state-managed animal, at least right now. And so there can be problems with low genetic diversity in grizzly bears, and we don't have all the information yet. So that's a great question.
Earlier you had stated that hunting areas don't necessarily correspond to genetic biodiversity. And so my question is, do you think it would be beneficial to conservation efforts if the number of animals harvested in a given region is more strongly correlated with the diversity in that region?
So the question is about the correlation between hunting areas and their genetics. And actually with bighorn sheep, I didn't describe that very well, but the genetics are correlating on a kind of coarse level with hunt areas. But I think that's something that Wyoming Game and Fish will examine more as we present more and more of the data to them. I do think, and I know that the wildlife biologists that are up on these things do care about that, and they do believe that genetically managed wildlife is a stronger way to go than if we don't include genetics in our management. And I'm getting a lot of really positive reception from this state, and I also did from California. So I think that's the future.
I'm sorry I missed it. Did you say that there were bighorn sheep population around here?
So right around here, there aren't, has anybody seen any bighorn sheep? Okay. Just to the north of here, up 191, to the Tetons, they used to be throughout the West, but not currently. And truthfully, I don't know if there's plans to translocate here. I don't know that there's the right habitat exactly right here. Does it?
That mountain goats in a little bit farther south from here, but I just...
So mountain goats are another issue actually up in the Tetons. The question is about mountain goats and their presence. Mountain goats were translocated back to the Tetons when they were almost basically extinct from that area, and they're exploding. They can carry those Pasteurella diseases. People love mountain goats, but now they're going population to population, nose to nose with the bighorn sheep. The mountain goats can carry that pathogen that kills bighorn sheep. Another dilemma. It's not easy. So, yeah.
When you were talking about the great gray owl, you mentioned that there's a population of less than 200. Are you saying less than 200 in Wyoming?
We don't know the exact number. The question is, how many do biologists, raptor biologists, think are in Wyoming? So the 1 to 200 is in California, in Yosemite. I've heard from Beth and the Teton Raptor Center biologists that we collaborate with that there may be 200 to 300. They don't have exact estimates yet. They're doing telemetry studies, our genetic studies, to try to estimate those numbers. But it's kind of low too. So a very coarse estimate. Sorry, I can't answer that right now. We're aiming to get a better estimate.
So if I could just try out my understanding from your talk, the
danger with genetic populations that are not very varied is that as they get small, they're not resistant to disease, and essentially they're not a robust population and could, as a population, disappear. What do you do to mitigate that? Is it more transplantation, or are there other ideas? Or?
So the question is, as populations get smaller, they get lower genetic diversity, what can we do to help prevent problems with that? So the first is, just like any conservation biologist, probably many of you think, is to avoid the problem in the first place. And the poster child for these problems, as I mentioned, is the Florida panther, a mountain lion, where they got the heart defects and other things. So the mitigation was to introduce fresh genetic material from Texas mountain lions. In the past, they were contiguous in their populations across the southeast. Well, it turns out that was a great idea. So the next generation of mountain lions between Texas and Florida panthers, the babies did not have heart defects and all the other problems. But recently our collaborators at CSU discovered there was a virus that was transmitted from those Texas panthers into the Florida panthers. So and if we move these animals around, it may be good on the short term, but we can't test for every disease they're going to carry or other genetic defects they might carry. I don't have an answer for that, except that we as a society need to prevent it from getting that small. And that's why Wyoming, I'm so excited to be here, because we have big wildlife populations, except for those great gray owls and a few others. Wyoming toads are tiny in population. But right now we have lots of pronghorn and mule deer. They're endangered, they're getting smaller. We have something we can do something about. So before it gets too small, and we have to do these millions of dollars of translocations and bringing in diseases when we don't even know we're doing it.
So is the problem an environmental problem in that we're separating populations, creating barriers between population movements? I mean, I know we've had Matt Calton speak Saturday to you several times about mule deer migration and pronghorn migration, and what he tries to use his work to influence is to prevent those barriers, migration, to find corridors, ways through private property that find owners that will allow migration where it's blocked in other places and so on. Is that a better approach, do you think?
So the question in the comment is that Dr. Matt Kaufman at the university, who we collaborate with on our ungulate studies, mentions there's important migratory pathways, and what's the main problem? And it is us, people. Too many people fragmenting habitats, cutting off those migration corridors, just like Dr. Kaufman says. And so one of the best things we can do is to prevent those fragmentation of the populations. Once they're fragmented, then that's that small population issue, low genetic diversity. So if we can act to protect those or encourage the migratory pathways, like the red desert to hoback for the pronghorn and the mule deer, and have private, public, collaboration to allow those animals to migrate through that they have for millennia, that's one of the best things we can do. Yeah.
What about the wolves? Is there much study being done genetically as to the introduction of the ones from Canada into the northwest corner of the state?
So the question is, what about wolves and genetics? And yes, some of my colleagues have worked for decades on wolf genetics. And so that's also being worked on. It's another case where it takes a lot of data, and now we're getting these big genomic data sets that we can learn about. And my colleagues are finding out more and more about their genetic diversity and the introduction of Canadian wolves to the system. So I don't have the answer for you, but I'd be happy to link up anybody with information about grizzly bears or wolves with the people who are the experts. Thank you.